Hello friends, good evening. Today we shall discuss about polytene chromosomes. In some organisms, certain tissues become polyploid during development. Such polyploidization process is probably a response to the needs for multiple copies of each chromosome and the genes that it carries. The process that produces such polyploid cells is called as endomitosis, which involves chromosome duplication followed by separation of sister chromatids. But there is no accompanying cell division. As a result, extra chromosome sets are accumulated within a single nucleus. Sometimes polyploidization occurs, but the sister chromatids fail to separate. In these cases, the duplicated chromosomes pile up next to each other, forming bundle of strands where the chromosomes are arranged in parallel. Such chromosomes are said to be polytene, means having many threads. Polytene chromosomes are multi-stranded and larger than the normal chromosomes. They are found in many species within the insect order Diptera, including flies and mosquitoes. This figure shows a single polytene chromosome. It consists of many sister chromatids which stay together in a parallel arrangement forming a thick bundle which is large enough to be seen under the light microscope. This is due to the fact that the chromosome undergoes several rounds of replication and the resulting sister chromatids fail to separate, so they remain together in a parallel arrangement forming a thick bundle which exhibit a pattern of dark bands and light interbands. Differential coiling along the length of a bundle cause variation in the density of chromatin. When dyes are applied to these chromosomes, the denser chromatin stay in more deeply. As a result, a pattern of dark bands and light bands is created. In the dark band region, because the chromatins are densely or tightly packed, so they take up more dyes. As a result, they become deeply colored or deeply stained. This differential staining along the length of a bundle creates a pattern of dark bands and light bands. Under certain conditions, the bands may exhibit chromosomal puffs, which are localized swellings of the chromosome. In the puff region, the chromatin have a more relaxed conformation and consequently a more open state. When radiolabeled uridine is added to Drosophila larvae briefly, radioactivity accumulates in the puff region, indicating that these are the site of active transcription. Such chromosomal puffs are also called as Balbiani rings. The most spectacular examples of polytene chromosomes are found in the salivary glands of Drosophila larvae. Each chromosome undergoes about 9 rounds of replication, thereby producing a total of about 500 copies in each cell. All copies pair tightly. As a result, a thick bundle is formed which can be seen under low magnification with a dissecting microscope. This figure shows the polytene chromosomes of Drosophila larvae. Out of various chromosomes, this is the fused chromosome 4 and that is the fused X chromosome. Remaining, there are 4 arms. These 2 arms belongs to the chromosome 2 and the remaining 2 arms belong to the chromosome 3. One arm is the left arm and the other is the right arm. All polytene chromosomes are thick bundles consisting of 2 homologous chromosomes which are paired up. Each homologous chromosome is composed of many copies of sister chromatids which fail to separate. As a result, all sister chromatids of the paired homologous chromosome stay together forming thick bundles. All these chromosomes exhibit a pattern of alternate dark and light bands. In this region, the two paired homologous chromosomes have been separated. The polytene chromosomes of Drosophila larvae exhibit two additional features. Number one, homologous polytene chromosomes pair up. Ordinarily, we think that pairing is a property of the meiotic chromosomes, but in many insect species, somatic chromosomes also pair. Because of this close pairing between homologous chromosomes, it is almost impossible to distinguish between them. Number 2. The centromere of all polytene chromosomes in Drosophila larvae merge to form a specific structure called chromocenter. As a result, various chromosomal arms seem to emanate out of the chromocenter. The chromocenter consists of heterochromatin. As a result, it remains almost unreplicated, whereas the chromosomal arms, which are banded, consist of euchromatin, so they are replicated. So friends, this is all about the polytene chromosomes. For more updates, please subscribe my YouTube channel, Sam Biology Class.